Okay, I guess we will get started. Wish I could make that go away because it's sharing the screen. Okay. So this week, we're going to go through the viral ecogenomics pipeline on both Cyverse and KBase. I will try to go through Cyverse a little faster because it will be easier to show you in KBase, like very specific details about each section than it is in what's it called? Than in Cyverse. So this is kind of the whole webinar. We're almost all the way to the end. This is me, and next week we'll go over. Well, I won't, but yeah, we'll move past all the viruses so that you can finally get a breather. Also last week, we went over the virus tools, OSC, IMG, Cyverse, and KBase. We didn't really do too much OSC. I think I had like 1 slide or 2 slides about it. I will make a note towards the end of the presentation about some other resources that are available to those who are at OSU, which I haven't checked the participant list, but should be some of you at least. Most of the resources that are available at all of these other areas are also available to you, and you can directly access them on OSC. These are the 2 pipelines we'll be going over. It's a little too tiny for you to read, but you can kind of see the flow of data, basically. Okay, so this is when I'm going to end the show. Okay, excellent. Let's see if we can move this away. There we go. So the first we'll start with is Cyverse. I have a fully featured protocols.io protocol basically for that. And I know you probably can't copy and paste this. So I actually wanted to show people protocols.io. I'm not signed in, so you don't have to be signed in for this. And I'll just type in Cyverse. See. Let's see, quality control, creating blast, preparing data. Let's see, where should we start? In fact, what did I call it? Sorry for that. That's a pipeline. Cyverse virus. Okay. So if you just type in Cyverse and iVirus, actually, I think if you type in iVirus, you'll get this too. But basically, we're going to be going to processing a viral metagenome using iVirus. It has been used by, at least this version, by 2.4 thousand viewers. And there's only a couple comments on this one. So we'll click on this. Okay, so this protocol is basically a collection of different protocols in Cyverse. It will go through, or we'll go through downloading the viral data, quality control of the reads using Chemomatic. Let's move this to the side so other people aren't seeing that. I'm still sharing my screen, right? My screen sharing. Assembling viral metagenomic data, clustering viral genomes. Let's see. Just want to make sure. Just going to share. It says I'm sharing this screen. Okay. As always, ask me a question if you cannot see my screen. It should be navigating Firefox, protocols.io. You should be able to see all of this. Please let me know if you can. And then prepare this data on Cyverse for vContact, visualize those, and then we have a bonus, which is mapping metagenomic data to our references so that you can estimate the abundance. So the first thing we want to do is open up Cyverse, cyverse.org. Usually go to de.cyverse.org. And we'll log in. Authentication. I've saved my thing for Cyverse, so I don't have to type it in. You don't have to create this. You can just follow along. I've used quite a bit of data. So the first thing we're going to do is navigate back to the protocols. Click on the downloading viral metagenomic data. Of course, it's going to warn me. This is all publicly available, so you don't have to log in. You don't have to sign up or any of that. And basically, this protocols.io has all of the steps. We went over this a little bit last week. All of the steps, some guidelines, materials, if this is a wet lab, some metadata about this. If you do cite it, please cite it in your papers. Here we have the metrics where you can see the number of views that people have had in the past version, at least for downloading file metagenomic data. So the first thing we're going to want to do is open up FAFSA dump. So we can actually navigate through. So this is on Cyverse again. Go to apps, go through, and what we want to do is look for high performance computing. Thankfully, this is saved, so everyone will be able to see this. Well, my internet could be slow. Well, Cyverse could be slow. Or we could have live issues. Could have live issues. Live issues. Okay. Let's try again. Okay, thank goodness. So we're gonna look up FAFSA dump. See if this will work. No results. Okay, let's just do FAFSA dump 1115 multi-file. Okay. Or 010. Let's go to the 010. Dump. Okay. So when you look through and navigate and find apps on Cyverse, you can search for them either the way I just did, or you could navigate through that very long list of apps, as I mentioned before. Oops, I'm looking under analysis, my bad. Okay, so it wasn't, let's navigate through some lists. Or not. Oh, you know why it's not here? I think it's under the general apps. Let's see what it is. It's FAFSA dump. Okay. This is just an example. Okay, okay. Back on track. Okay, so every app looks like this in Cyverse. It has the analysis info, the parameters, and then you review those parameters and run it. You can call it whatever you want, but kind of be descriptive. I usually have the analysis name and then some type of date or time, or all of that's kind of kept in the DE, so you can review it later, but it's often easier depending on what kind of paper or protocol or whatever you're working on to give it some type of useful name. Next are the parameters, and for this you could pretty much choose whatever accession you want. I will just type this in and I'll just start the run, but I'll kind of quickly go through this so that we don't have to spend hours on each single step. It's 771003, and it was what, ER? Yes. Next. So these are parameters. There's only one parameter here, and I could go ahead and launch this analysis. In fact, let's see. Okay, so I'm actually running a job on the HPC that's associated with Cyverse. So I could be using dozens of cores right now. I probably aren't, because the developer didn't probably set this to multiple cores. And so this app was just submitted. It's running, it's probably in the queue right now. And you can watch as it gets through the processing stages. I don't know how else. Okay. And if you go to analyses, okay, analyses, it will pop up with, okay, we've just started this. It's submitted. If we wanted to, we could relaunch it. If we didn't like the parameters that we used, we could get other details about it. And then once it's done, you can actually click on the app, and then it will give you some information about it. Okay. So yeah, I do a lot of development on Cyverse. And so, oops, let's go here. And so that once that's done and you click on it, you'll be able to actually click on a directory that's generated, and that will take you to the output folder. And let's see if I have SRA, FAFSQ dump, bam. Okay, of course, because I cleaned it up. Okay, so you'll run the job, and let's see. So because this is live and not everything will work, I will use this as a teaching moment. And if you navigate to Cyverse and go to the community data under the data section, I have the entire pipeline. I probably should have opened with that. Under virus, and then I have the, should be under example data. Example data. Okay. So all of the apps, all of the tools that I'm using here actually have input data and the output from each one of those. And let's say, I don't think I've got FAFSQ dump here. The reason we don't have FAFSQ dump, let's actually look at Viromics. This is the next step that we're going to talk about. Okay, so if we were to download this ERR file, what's going to happen is it's going to create this directory. I know this is kind of an older version of it, with two FAFSA files, FAFSQ files, and they're going to be, moderately small. And let's see, let's just assume that they're the ocean sampling day reads or whatever. you'll get them and you'll have two files that have the errors or the outputs or anything that was generated from the run. And let me actually open up the questions in case anyone's having. Okay. And let's see. Okay, so that's what we'll have as outputs. The next step after we've downloaded this data, let's see what the next step is. Next step is to quality control those reads. So the next app is going to be Chemomatic. Let me stop switching between all of this, because it will probably be easier just to show you. Geez, here. A lot of analyses, apps, Chemomatic. And I don't want to belabor the point, but basically, this whole process, you go to Cyverse, you look for your app, click on an app, you go through, you put in some parameters. Oh, that was for the other tools. In fact, let's go to the OSU Viromics workshop. Okay. So, yes. So we're going to be running Chemomatic. So I just went to the iVirus 2019 OSU Viromics workshop. I've run this pipeline a couple different times, and I've put it on in a whole bunch of different places, depending on what the community needed. And so for Chemomatic, you can click on this data, and it will give you all the input data, the output data. For this, I think some of these examples we took, I'm not sure actually where we took these examples from, but it's all public data, of course, because it was downloaded using the SRA. And you can click on read files that you might need. Read files here. Of course, it's going to be a struggle. QC and assembly, Chemomatic, and then click on the second. I'm not going to run all of these. And then, of course, we have an Illumina adapter file. I don't know why it's not saving these. I'm going to speed up pretty soon here, just so that you can kind of see. I think this was clearly, I must have used Nextera. And then what we didn't see in the FAFSA dump was some of the parameters. And these parameters are kind of all laid out for you in the protocols. So you can actually navigate to iVirus example data, Chemomatic input reads. This is what I had actually navigated to before. And this will go through reads file. You can use any forward or unpaired read you want. You can use the reverse or the mate pair, the Illumina adapter file, or whatever other adapter primers that you want. You can upload your own if those aren't working for you. And then afterward, you can call these outputs whatever you want, window sizes. These are all defaults. So someone last week was discussing the parameters that you need for Chemomatic. Some of the parameters that Chemomatic has are slightly tweaked here. I think I've got the minimum read length pretty high here. It could be 30 or 35. I probably wouldn't trust anything below that. I think for this example, I wanted to keep it kind of standard at 50. But the leading quality, the seed mismatches, and all of this stuff, you can generally go with the defaults that I've set here. These aren't exactly Trimomatics defaults, so just keep those in mind, because if you download the app or the tool, I'm sorry, and you run it, and then you run this on Cyverse, you're going to get minuscule differences. I doubt those differences are going to be significant, but they will not be exactly the same. One other thing to note is that there's going to be 2 output files for every single input read you have. And so Trimmomatic will output both the paired or unpaired, at least for the paired end data you have. So if you put in a forward and reverse pair of reads, you're going to get the forward reads that survive with a pair, the reverse reads that survive that also have a pair, that's 2. You're going to have the reverse reads that don't have a pair anymore, so its mate perished, didn't survive, and then you're going to have it for the forward reads as well. And then we can press next and then launch analysis. I'm not going to do that because this is going to take 10, 15, 20 minutes. Before someone asks, there are a couple other quality control tools that you can use besides Trimmomatic on Cyverse. I haven't gone through them, but last week I went over and gave a couple examples. You can just, type them in here. I don't think the BB tools are in here. BB tools. No, BB. I do not believe so. Okay. I think cut adapt is in here. Or there's so many apps that it now takes forever to go through them. I'm fairly certain that there's at least a handful of other tools besides Trimmomatic that you can use. Unable to search. Okay. Okay. So you'll type all of those in, and you're going to get, wow, this is really low quality. You're going to get at least those 4 output files that I was mentioning. You're going to have the forward and the reverse that have their pairs surviving, and then you're going to have their forward and reverse where their pairs didn't survive. So sad sounding. Okay, so after you do this, after you do the quality control of reads, we're going to actually assemble that data. So for this, we want to use spades or meta spades. If you've ever used this kind of, let's see, spades, is this going to work for us? Yes, spades. Okay. So same intro, overall, go to parameters. You can put in a number of different paired end libraries if you'd like. You can put in single end libraries as well, PacBio, Nanopore, Sanger. 3.12 is actually relatively recent. I'm sure there's quite a few other versions in here. If you really need like 3.15, which I think is the latest, you can always ask Cyverse to see if they can do something about updating it to the more recent version. But keep in mind, like not every single, they're not going to be able to accommodate every single release. So do be mindful when you're requesting that. So we can do the same thing that we did before. I do have examples, basically as you've seen. So the output from that, from the Trimmomatic, actually we could go through FastQC. I probably should have done that first, but we'll do that in KBase as well. Will be generated, and you can just rename those outputs. You don't have to if you don't want to. Let's click on output for the first pair. like I know what these reads are, and so I didn't have a creative name, so that's kind of my bad. And this is the output from Trimmomatic, i.e. the input for spades or metaspades. I should give you some details. We don't have PacBio, Nanopore, Sanger reads, but you could put all of those in here. You probably want to know some details like the parameters. For stuff like spades, it's pretty common to use these k-mers, 21, 33, 55, 77, 99, and 121. That's kind of a good default set of k-mers to use. We don't have single cell data. This isn't plasma data. This is metagenomic data. And we don't have any RNA data right here. But Spades does have an RNA Spades version of it. So if you do meta flag for Spades, it enables the meta Spades kind of set of algorithms. If you do RNA, it will run RNA Spades. Plasmid, as you can take, it will run plasma Spades. I don't think there's a single cell Spades, but it does. Spades will adjust its parameters to basically internally to assume that most of the data is coming from the same cell. And then next, and then we can review all of the options. It shouldn't be too many options, too many questions about that. I have multiple monitors here, so if you see me looking around like a crazy person, that's why. Okay, so we do have one question. Let me move this window a little closer so you know what I was getting inside profile. If you want to search for DNA viruses and want to exclude host DNA using Bowtie, should we do that after Trimmomatic and before Spades? Let's see, search for DNA viruses, exclude host DNA. I would actually run Trimmomatic. I personally would run Trimmomatic first. And here's the reason. So when you're mapping with BWA or Bowtie, those adapters and primers are going to interfere with the mapping algorithm. So of either BWA or Bowtie or whatever other tool you want to use. And so if you want to ensure that you're trimming off all of the sequencing data information, primers, primers, and adapters, then yeah, that's what I would do first. So get your data, clean it up using Trimmomatic or whatever other trimming tool you want, and then map out your host DNA, and then assemble. Yes, and then assemble. Okay. And of course, obligatory go through protocols. These are the same data that I used before. This is an old version of the protocol. So one great thing you can do on protocols, if I were logged in, I could actually add a comment and say, hey, oops, I gotta sign in. Okay, don't yell at me. I could write, if I signed in, author, why do you have these 3 k-mers? I thought that the best practices guidelines said that you should have a few more k-mers than that, or to improve it. And then someone like myself would come by and respond to that. Oh, that was to the previous question. Previous step. Respond with, oh, yes, I will update the protocol to make sure that it has the latest and greatest versions. And then launch. And then what you should get in this really low quality example, let's actually go to data for a second. Just data. Eating at my soul, navigating through everything. 2019 That your Viromics workshop. QC and assembly. Output. Okay. This is actually the output directory that you would get. That is what's represented in this tiny little figure, except I've kind of cleaned it up so that you don't have all the other Kmer and temporary junk to clean things up. And the files that we are interested in are the contigs.fafsta, because this will have all of your assembled data right there. There's a couple other files that could be useful to you, such as like the assembly graph. And unfortunately, Cyverse doesn't have, I think, any tools that will look through the assembly graph, which is basically a visual representation of how the assembly assembled, how the assembler put together the reads to form the contigs. And there's nothing else really major to mention about metaspades. We're still in the this isn't so viral part of the ecogenomics pipeline. We need to really improve that quality. Or maybe that's for logged in users. Okay, so after we've done that, we can go through and cluster these virus genomes, or we could immediately identify these sequences using VirSorter. I typically, actually these steps are out of order. So after we assemble the metagenomic data, I need to check on this. We need to identify the viral sequences using VirSorter. And this is an old version of VirSorter, so there's an updated version. Let's look for this. Apps, VirSorter 2. I definitely clicked on an old version of that. Apps, VirSorter 2. Okay, so I've now clicked on VirSorter 2. This is when we're actually getting to real science. Real science. Does Trimomatic have the Illumina sequences already incorporated into it? No, Trimomatic does not. You will need to select them either from a file that you've uploaded, a primary or adaptive file that you've uploaded, or you can use the adaptive files that are available through the iVirus folder that I've keep on navigating to. Those should have most, if not all, of the adapters that Trimomatic uses. So that was good. What software can create assembly graphs from assemblies? So the assembly graphs are generally generated by the assemblers themselves. I am not aware of any tools. I know there has to be a tool that exists though, that will generate the assembly graph from, some intermediate file of the assembler. I don't know. I think Spades is really the big name that generates the assembly graph. If someone else has any other assemblers they think or know of that generate the assembly graphs, do let me know. I often use the assembly graphs to kind of visually look at the quality of the assembly without needing to use some of the other assembly, like Quast or MetaQuast, that I don't go over in here, but that's definitely a tool that I use to look at the overall quality. Okay. And okay. So for VirSorter, getting into real science now. Inputs, this will be the contigs file from the assembly. Let's see. Should have the assembly output here. I'm not sure why there's no assembly output here. Let's go to example data. And let's look for Virosorter 2. Okay, test. Okay. So this is iVirus example data, Virosorter 2, etc. So the input is basically any FAFSA file that you'd like to use. It could be compressed or not compressed. This should be your cleaned up raw assembly, if you will. It could be microbial data, it could be viral data, it could be any type of data you want to identify viruses from. For the parameters, if you have TRAM-V, for example, you want to flag this option. If you want to exclude short sequences, you could flag this option. There is a protocol on protocols.io that go through all of the SOP for running Virosorter 2. In fact, let's go to that. Sorry, I'm jumping around. Once we leave Cyverse, it will be much less so. Virosorter 2. Okay. So there's a sequence identification protocol that actually goes through all of the options for running Virosorter. And it's pretty detailed. And so there's this one, and then there's also, I think there's getting started. Yeah, it goes through a couple of these options on how to install it, running it, output files, training the customized classifiers that is way beyond the scope of this discussion. But suffice it to say, there's only really a couple options that you probably need to change. The first is the viral groups to include. Virosorter 2 is benchmarked against a number of different virus types, such as the large viruses, RNA viruses, single-stranded DNA viruses, double-stranded DNA phages. And you can add or remove different groups. If I don't have any RNA data, there's no need to try to look for RNA viruses. All of those were the 2, 4, 5, 6 viral groups that Virosorter 2 has been trained with. And so you're not going to get any more than that. I sometimes, if I'm just running Virosorter 2 on a large data set and I don't want to have to kind of manually look through any of the results, I've actually selected the only output high confidence viral sequences. And these should be, have a very low false positive rate, very high, true positive rate. And the max score, I just wanted to say that you can generate this flag manually by adjusting these options. So, for example, if you only output high confidence viral contigs, its max score is 0.9, or the max score is 0.7 and the hallmark is greater than 1. Some of these have come from the SOP that I was going through, that have been identified through the SOP, and then a lot of internal work that the lab has done. And so those should give you a fairly high confidence that the sequences generated by this automatically are viruses. So you could also produce the same kind of results if you'd ask, okay, I need a hallmark gene. So you need to have at least a hallmark, and then adjusting my score, it's right below this, to 0.7. And then I'd be able to get, this value. Or maybe I want, maybe my virus doesn't have a hallmark gene. Or the viruses that I'm interested in maybe don't have any, known hallmark genes. And so I would want the confidence to be 0.9, the score cutoff to be 0.9. And then I would have to run it again and then combine those. So you can change them, you can adjust this, you can run VirSorter 2 a couple times. You should probably start with a lower cutoff, and then if you want to go through and manually adjust them afterward, and manually filter them afterward if you'd like. Hallmarks genes, yes. So if you do have smaller sequences, so RNA viruses are the big ones. You can have single-stranded DNA viruses. They can be shorter than 5 kb, which VirSorter 2 considers short sequences. And so you might want to clean out any of the small quote-unquote junk in your data set by not having VirSorter work on those or try to process those by just discarding all of your short sequences. And you'll see that I actually do that in KBase. However, you might want to keep those. For example, if you want an RNA virus that has the hallmark RDRP gene. And so if you have a 3 kb virus that has a full-length RDRP, you want to keep that because you're quite confident that that genome is a virus. And so you'd want to flag that on. You don't have to, especially if you don't have any RNA virus data that you're working with, or you're not interested in the really tiny viruses that, tiny viral genomes that might, make VirSorter 2 take longer to run. And you're really not interested in something really tiny like that, because another reason is that downstream apps and tools might not be able to work out something less than 3 kb. Only be annotated by sequencing cDNA. So the question is, will RNA viruses only be annotated by sequencing cDNA? Annotated, no, you can put in, if you had direct RNA sequencing or, through cDNA, VirSorter 2 would identify them in both cases. If, I guess I kind of answered that. If you mean by if you can only get RNA data through cDNA synthesis, there are alternative ways of doing direct RNA sequencing, and that would still be workable here, but the vast majority of RNA sequence data that you're getting is through a cDNA intermediate. Done. Okay. So let's assume we've now identified your viral sequences using VirSorter. That's when we get to kind of the part of the pipeline where there's a lot of stuff we can do. And I guess the phrase, the world's your oyster. But you can annotate genes. You can identify the taxonomy using vContact2. You can do population ecology using Metapop. You can do some read mapping using Bowtie2 and Bowtie2 batch. You can do protein clustering. There's a whole bunch of different apps that you can then run on it. And two things I actually wanted to show you was, okay, so one thing that people always want to do is map their data in order to get kind of type of abundance. How abundant is my particular sequence in these set of samples? And so let's assume, we'll just stay with protocol so I don't keep on switching back and forth. Let's assume you've done your VirSorter 2, you've identified your viruses. Now you want to figure out, okay, how abundant are these viruses in my global ocean data set? And so you would navigate through, and you can do this yourself as well. Go to the discovery environment, click on and find Bowtie batch. And then we're going to go through and select the inputs. So the inputs that you want to select are, of course, your viruses that you're interested in identifying, their abundances. And then the next thing you'd want to add is a directory that has all of your read data. So these are your forward reads, your reverse reads, your interleaved reads, etc. And once you've selected that, let's actually keep on telling. I just want to show you kind of the new type batch. Let me just show you the new interface. Cyverse just upgraded their interface. And up until like a couple months ago, you could still use the old interface, but I don't think you can use the old one anymore. And so this is a reference file with your sequences, a directory of the reads. Yeah, it's going to yell at me. And so let's actually look at batch. Like, so all of these examples, you can run, they're runnable. This is the ocean sampling day data set. And so you basically click on this read directory and be like, I want all of the reads. I want to map all of the reads. You put in what kind of parameters they are, what kind of reads you are, what do you have paired and unpaired. I think I've got only paired. You can keep the SAM files if you wanted. Parameters for merging your data if you want to merge them. You don't need to. All of these, the defaults are fine here. And then you'd go through and press next, 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 and run. So, and I go through a little bit of detail on what each of these options mean. The Leverstein distance is kind of the distance that I use between the read names to identify, should reads be paired to each other or not, or how to identify what reads are mapped to paired to each other, sorry. And you might have to change that if it gives you weird results, or if there's actually be a warning that says, hey, there's something that looks off. We would still be able to run it, but one of your read pairs had 10,000 reads and the other one had 50,000. Doesn't look like they're matching. But you can adjust the distance and that will get fixed. And so once that's done, you're going to get a whole bunch of SAM files. One SAM file for each of the read pairs or interleave files that you generated. And let's go back to data. Oops, that's all my data. And actually, let's just get back to here. You'll get a directory that has all of the reads. BAM files, SAM files. And I think right here, I went through and showed people what the log file kind of looks like. And this is actually where you'd see the warning if something didn't map correctly. But it says there's no problem apparently with it. So this is good. And you're left with a couple BAM files. And then afterward, you'll open this tool called read to ref mapper. Read to ref mapper. Read to ref mapper that is not data. Read to ref mapper. Do I have to be really specific? No, I don't. Okay, excellent. This is going to be the directory that we just generated from Bowtie Batch. You can use a metagenome sizes file. This will help normalize the reads that you have in your data set. And that's useful for when you have different sequencing depths. So if you have 5 metagenomes that have, 100 megabases of sequencing, And then you have another metagenome that has 5 megabases of sequencing. If you notice that your virus is 10 times less abundant in the 5 megabase data set, it's likely not due to a change in the abundance of the actual virus. Any change or difference you would see is actually due to the sequencing depth or effort that you put in there. And so what this metagenome sizes file does is that if you've calculated the size of your metagenomes, you can put those in this CSV file, and it will adjust your data set for you. It will basically divide out by the largest data set and then multiply by some factor to get them all within the relative, same amounts, so that you can compare your abundance from one sample to another. And then the next part of the parameters here, the read percent coverage, the percent ID, these are based on, slightly based on the defaults. We probably should have 95 and probably 85% coverage to make them agree with the MUVIGS standards. I think last time it was 80. Could have been 8. I think it was 85 actually, and 95% identity. These other defaults are just fine. And what this will do is once you press next and run, I should keep on going back to that. Metagenome file, I've got the example files, etc., the example directories, etc. Adjust, results, scroll through. You're going to get a coverage table. And unfortunately, there's no good visualization tools in Cyverse, at least on the metagenomic side. And it will generate this table for you that has the relative abundance of each of your genomes or contigs in this particular data set. And yes, so there's the unadjusted, so this is the raw coverage table. And then there'll be a adjusted coverage table that actually takes into consideration the mapping file. And the reason for both of these files is if you don't have a mapping file, of course, you're not going to generate it, the adjusted coverage table. And then 2, you might want the raw data for some type of other analysis. And so, keeping that there is kind of useful for various users. And then if you run that, you'll get this kind of heat map. Now, this is a far cry from what you might see in Science or Nature, but this is only like a very, an example, kind of like a gross overview of what the data set looks like. I would strongly advise to not use this in a Nature or a Science level publication, and instead download these files, the adjusted coverage table or the raw one, pull it into Python, pull it into your favorite heat map tool, and generate a much better figure. You can actually add some other attributes, metadata to this, and really make it kind of Science or Nature worthy. Okay. Mapping. So vContact, I'm actually going to save vContact for KBase. And then clustering viral genomes. Okay, so something else that, and I won't spend too long on this either. Something that a lot of people want to do and to kind of satisfy the MUVIG best practices is once you've identified your viruses, you want to make sure that each virus sequence is either a species or a population, etc. Because what you don't want to do is think you have 2, 3, 4, or 5 genomes of the same virus that are really only just, resulting from the assembly or assembler not being able to put those genomes together fully. And it will affect your abundances because you're going to have, like, Bowtie or BWA is going to have a hard time if you have 5 of the same sequences just out that you're mapping against. And so you want to make sure that when you're talking about a single unit, that you want it to be the same species or the same viral population, or basically the same genome. And cluster genomes is one of those tools. You can use any other tool that you want. You can use DREP. You can use, I'm forgetting the name right now, but basically almost any clustering tool that you could think of could be suitable for virus data. And so you probably want to set the coverages like 95 or 80. That's kind of, I think that was the MUVIG standard for clustering these genomes together. And then what you'll get is basically, and those are the only 2 options. You'll get a seeds.fna file. And this file is going to be the one that you want to use for subsequent, data analysis steps. So yeah, that is the metagenome size units. So yes, it can be in either base pairs or in reads. I use base pairs by standards, and I use the quality controlled results at the end of Chemomatic, because I don't want to include the adapters and the primers and all that other sequence prep bases, basically. Okay. And that is pretty much most of the ecogenomics pipeline I have for, oops, it's not, for Cyverse. I don't have protocols for Metapop enabled quite yet, but the app is available in the Discover environment as well. Metapop. So Metapop is a population ecology tool that you can often use. Generally, you could use it after you've identified your viruses. I have it here that you pass vContact2 data kind of sort of through. And that's kind of a misnomer. It's basically the direction is after you've done viral identification, you can go through all of these steps. And it consumes the same kind of inputs that Bowtie and Bowtie batch used. So, or that Bowtie batch generated. So it consumes a directory of BAM files. It also has its own normalization file that you can use. This will be calculated in Metapop. And it also uses those reference genomes as well that you used in Bowtie and read to ref mapper. I do also have inputs for this as well. Example data, Metapop, BAM directory should look something like this. Let's see. Predicted, that's optional. FAFSA files. Also, we should navigate through there. It could be this. It could be any FAFSA file, normalization file. All these defaults are like, you don't have to click on anything, basically. And it will take you through, I really should have an example of this. But once that's done, it will generate, in fact, we can cheat a little. Think of it. Okay. Actually, we'll skip on that to go through. So yeah, the normalization file is all the same. And it will generate several files that have all of the all of the diversity statistics you can imagine. I believe it does alpha diversity, it does microdiversity, macrodiversity. It will visualize like theta and pi and a few others. I'm really out of my depth here when talking about, like population ecology. But if you're interested in Metapop, definitely check out the paper. And this is the app. This is the tool that you can use in order to generate all of those crazy diversity statistics. And I think that's it for Cyverse. I know there isn't, I don't feel like, God, I've been speaking for an hour and I feel like I haven't even talked about anything. Goodness. So if, even if it's not in this kind of protocol here, it's definitely, you should search through the apps, sorry, and look through and see if we've added any additional apps or tools. You can check out the iVirus website to see the latest and greatest that we've added here. There might be some other tools here. I don't know if you can really search as well through virus stuff. Okay, so you can have filter virus associated with from host reads. Oh yeah, MARVD, my goodness. So you can also identify archaeoviruses from your sequencing data. Probably you want to use MARVD2. There's a genome annotation tool for papillom virus genomes. And that might be it. One of the reasons why there's only a couple tools is because a lot of people aren't interested in viruses, unfortunately. And so you have these giant cyber infrastructures that are often focused on plants, humans, for obvious reasons, and other kind of organisms that kind of get more of the limelight. But a lot of the efforts of iVirus and a couple other collaborators, we've been trying to get more virus tools out there. So, yes. So the next thing I wanted to actually focus on, I'll give us all like a second to breathe. Okay, so K-base is kind of the next kind of step. And there's two protocols here that I've done. One is an older one that's about 6 months old, and absolutely everything has been processed in it, but it hasn't gone through the entire, like the new version. For example, this version uses the old vir sorter, and the new one uses the new vir sorter. The old one didn't have check V, the new one had check V. And so these two protocols, in fact, I don't know if I can really like copy and paste these for you. Keybase doesn't have the easiest way to look through. So let's go to Keybase.us. Let's log in. Okay. So you should be able to go through and go to narratives, public, let's type in virus. Huh. Okay, yes. And it's the viral analysis end to end. And if we click on that, it will take us to this narrative. And this narrative is kind of a slightly older version of this other one that I want to show you. And the difference is that I still have one app that's still running. KBase actually had maintenance yesterday, and so it kind of killed my running job. So I apologize that it can't be like 100.0% finished. But what this pipeline does is you can basically scroll through. This is like a whole lab notebook. Actually, let me stop for a second and address a couple questions. If we want to search for DNA viruses and exclude host DNA using Bowtie, should we do that after Traumatic and before? Oh, I think I already answered that. I apologize. Done. 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 Okay, so what's the difference between Cyberus and KBase? Well, technically speaking, Cyberus is a cyber infrastructure, and KBase is a platform, but that's kind of a cheap answer. Cyberus is, to put it a little bit more succinctly, Cyberus is basically a very, very large HPC computational resources that has various services that the user can access. So one service is the discovery environment, which is what we were looking at. Another service is all of the data storage. So you don't even have to use Cyberus like we are using apps. You can basically upload data to Cyberus and use it to store your data. You can use it to run analysis on your local machine, upload it to Cyberus, and then use that almost as a storage place. You can use it. There's another kind of window for processing images, and all of that is kind of under the banner of Cyberus. And there's a whole bunch of others. There's like the DNA subway, which basically is kind of a traditional, let's process a DNA data set that's not designed for viruses at all, which is why I haven't discussed it. And there's a few others out there that I don't really use too much. They also have like a version that runs in the cloud as well, or for running cloud-based analysis. And yeah, those are all different components of Cyberus. KBase is a much more focused, much more, I want to say more laboratory-oriented view of data analysis, if you will. So one is kind of the more old school traditional. I'm going to have files and folders, directories. I'm going to be running things on an HPC and let the user have as much possible control. KBase, on the other hand, has really kind of narrowed the focus dramatically and organized everything basically by what data you have, and you don't have really option of how the data is stored. You can only upload data and download data. You don't have like all the intermediate files available. Only if the developer is letting you download the data are you actually going to be able to download it. So for example, the Metaspades assembly graph file in Cyberus, you'd have access to all of that. You'd have access to all of the intermediate files taking up hundreds of gigabytes of data, disk space. On KBase, no, you only have that one output file. and you can't even download that output file as like that raw file from Spades. You'll basically save, KBase will save it into an object, an assembly object, and then you can download or convert that assembly object into a FAFSTA. And so KBase has like absolute total kind of control over the data objects, or like kind of like every step of the pipeline. And that's kind of in stark contrast to what Cyverse offers. That said, KBase, because they have that kind of control over the data object, allows you to do kind of some pretty incredible things in this kind of, in the framework that they've designed. And I think that's going to be really apparent when I go through the KBase version of this. So yes, so yeah, I definitely, I hope this will answer your question, anonymous attendees. So let's see, thanks, okay. Okay, this other pipeline that I'm actually working on, which is the exact same thing, is, oops, let's go back here. I hate having to do this. So if you do to kbase.us, 126.189, and then -25. I don't actually have another way of looking for it. It is public and you can find it. It just doesn't pop up at the top for some reason. I think it's because I just now did it not too long ago. But if you type in that kbase.us and 126.189-/25. This is a narrative that has all of the data, has all the references and citations and whatnot that you're looking at, basically, but you're not in KBase, you're not in a narrative. And so you can basically follow along with all of the data and everything without even like logging into KBase. This is publicly available. And this is kind of a more fully featured version of the protocols, the Cyverse protocols version, simply because because of how KBase structures their data. And so this is the same thing that I've been discussing before. And also another thing with KBase is kind of clear here, I've been meandering on for too long, is the fact that everything is more visually oriented. And so these are graphics that I can upload that you can't do in Cyverse, of this pipeline, of this process, of this protocol, basically. And in here, it's basically a tutorial. This will take you through the whole process in KBase. So the tutorial objectives: assemble this viral metagenome using raw reads, identify them, and then classify them. This sounds very, very familiar. I have the background so that if you want to study viruses, these are some really cool statistics that people don't often consider. Like they lyse between 20 to 40% of ocean microbes every single day. They think that around 8% of the human genome is viral, the most biological abundant entities, basically. And so there's a lot more information you kind of add to this, the context. And if you're running an analysis, you want to publish it in like Science or Nature or something, you can create this protocol with your real data, your actual data, and everything will be available to every single person when you're publishing that. The same can be said for Cyverse, but it's going to be in a lot of different steps, and you can't use like one URL for Cyverse, unlike you can with KBase. So we'll import the read data. This is the exact same thing. So you now, after looking through kind of the old school Cyverse, now you get to see the different way of looking at stuff. So in here, I've uploaded images from various references, citations and whatnot, so that you can actually click. Is this going to take a new thing? Yes. No, accept all cookies, whatever. You can actually look through. Of course, I need to sign in. But yeah, you can go through all these citations and read about the data that we're downloading here. And so we're going to download this site. And I have some ways of navigating through. So basically, we want to go to the NIH, the SRA, go to data analysis, and then basically copy and paste this link. I actually take you through. I like to hold people's hands because sometimes when I'm trying to go through other people's protocols, they always miss, they miss that one click or that one step or that one line. And that just like, I don't see it. Where is it? And it's hard to make that kind of connection. And so I try to literally lay out every single thing that you need to do here. So click on data access, copy and paste this line. And then I have it here, but we can basically search for this, and I'm like SRA. And so we want to upload data. So we want to click on this import reads from the web. And it will open up. In fact, I can just do it right now. Open up this. And it will pop up with a, oh, do you have a direct download link? Yes. SRA, and then type in all of this information. The cool thing is that because I've shared this narrative and made it public, everyone has access to everything that I did. So if I can look at the configuration of when I ran this. I've copy and pasted that SRA URL. This is the read object name. I'll get that in a second, but sequencing technology and stuff like that. You can get information about that, how to use it, and whatnot. Job status. It queued for 10 seconds and ran for 2 hours. It takes a lot of time to run stuff from the SRA. And then a result, and it created an object, a paired end library object. And this is the hardest thing to wrap definitely for a developer, but the hardest thing for maybe the public or users to wrap their brain around is that there's no files that you're handling. You're passing in objects. So if you have a paired end library object, you can only use apps that accept paired end libraries. So Spades can use paired end libraries because it's an assembler. Bowtie Batch could use these, could use paired end libraries because it uses reads. However, if you wanted to run VirSorter, that can't use paired end libraries because it doesn't make sense for you to be predicting virus sequences on those raw reads. Of course, there are tools out there that kind of classify stuff, but that's besides the point. The point is that the app isn't designed to, VirSorter isn't designed to use reads as an input. And the developer, so I only have certain inputs that VirSorter can use. Paired end libraries aren't one of them. And so you never find yourself in a situation where an app, you're putting input data into an app that can't accept it. In Cyverse, you could. I could say FASTA file, and you have no idea if I'm talking about reads or general contigs or virus contigs or any of that. And in Kbase, I don't want to say can't, but it's much, much harder to make that same mistake. Okay, so we downloaded, that's my little Cyverse Kbase spiel. So overview of what we did, the stats. Kbase is also very nice to us. Tells us how many reads are in there, how many bases, the statistics, the GC base percentage, and all of that. What it also does is generates that object for us, that SRA reads thing. Where are we? Okay. And in the data directory over here, data tab, we can see that we have, where are you? ERR, this paired end library. If you click down, you can see some of the statistics about the quality of these. And then if we click on this, I won't do this to every object because we'll be here all day. Oh, okay, it's not this one. Sorry. Apologize. Oops, that cell. You can get all those read statistics. That's actually what I was going to show you, but it's actually already in the in the report. Okay, so after that, we're going to assess this read quality. And this is something I skipped in Cyverse, but it's the exact same thing. You go into the app, you're searching for that. And so we want to search for quality. You can kind of just spoil the view, FAFSQC, read processing, click on assessing this. In fact, they've just updated it. Wow. 11.5. And I just did this like, actually, no, I didn't do this on October 27. Oh, I reused this app. Okay, gotcha. So yeah, 11.9 versus 11.5. And basically, since we're viewing the configuration, I basically put in that read file, that paired end library, and then there's no parameters because it doesn't have any parameters to speak of. Job status, it ran for 31 minutes. And then the result is the actual FAFSQC report. So instead of going through Cyverse and navigating to the output directory and all of that, it's actually generated for you straight visually, straight in your window. Going over the FAFSQC report in detail is also unfortunately beyond our scope, but suffice to say, you want most of your reads to be in the green. And as you can see, there's a few reads that are, a couple reads that are in the medium quality. And if you've never seen this before, this is basically looking at the quality scores of every single read. Actually, it takes a sampling, I believe, and their position. So this is the 5' or the left part of the reads on average, summed up, well, averaged across all the reads in your data set. And you can see that the read quality in the middle of the read is the highest quality right here. And then as you get towards the end of the read, the quality tapers. And then of course you can have statistics about, what kind of sequence duplication do we have, the content, the GC content per tile quality. It's like, oh, there's low quality in that particular group. Per sequence GC content, what kind of distribution do we have? You can often tell if you have multiple different large subpopulations in here by looking at this. The GC content, sequence length distribution, like how long are our reads, duplication levels. So it looks pretty good. And then, yeah, I could spend all day looking at this. Just making sure we don't have too many questions. Okay. So after you've assessed this quality, and I go through the detail on like why you'd want to do that. Basically, you want to see what your quality of data is before you even do anything. Like, am I even starting with something good? Because if it's bad, like really bad, like everything's in the red or almost everything's in the red and there's a couple things in the orange, then you might want to look at how did you prepare your data? How did you get to this point? And so, yeah, you could get away. I think I say that here. You could get away with that because it's mostly really good data. But yeah, let's trim it with Trim-O-Matic. And so we will, of course, we're looking for read processing. In fact, let's Let's change the category. Actually, we're already at category. Let's look at read processing. There's 20 apps for read processing. So you can read error correction. We just did FastQC, cut adapt, filter out low complexity reads. That's only importing the data. Join overlapping mate pairs with FastQ join. That might be useful for a couple of people depending on your output analysis or your downstream analyses. Randomly subsample reads. Maybe if you have too many, your data set's too large. The BB tools filtering pipeline is here. And then finally trim reads with Trim-O-Matic, which is what we did. And to answer the question from earlier, we actually, in fact, let's just grab this. Reads object. So this is it filtering. It's looking through the data I have and saying, what kind of reads do you have available? So I could use the paired in library. I could use the trimmed paired. I could use the unpaired data. In fact, this is a little spoiler because these are actually the trim data from the earlier Trim-O-Matic run. And so let's use our raw read data. We want to enable the clipping. We have all of the adapters available already there. All of these parameters are the defaults in KBase. You can keep them as is. And then we want to title this, usually I use the, you can put reads.t. I often use .t for trimmed, or .qc for it's been QC'd. I used to put like Trim-O-Matic and stuff like that in there, but usually when I'm running my pipelines, I stick with one of them. I either use Bolduc or Trim-O-Matic to clean up my my reads, and I just stay with it in that whole pipeline. And then I go through and re-QC, assess the quality again. And now you can see that some of those X's and a couple of those exclamation points are now far fewer in here. And if we look at the quality, oh my goodness, absolutely everything's, well, don't make me a liar. Almost everything is in the green. And this is really good data already. So we were only marginally improving, basically getting rid of some of the junk. Those are probably primers at the front end, and just a byproduct of the Illumina sequencing process. So this is great. Good data. And I'm sure someone's going to ask, like, how can you tell? How do I know my data is good? It's a very long, it'll be a long-winded answer, but I can summarize it by saying, just try to get most of your data into this green. Because these are basically confidence or like probability that your base is incorrect. But once you start hitting like 30 plus, the chances of that being a bad base call is so minuscule that it isn't going to affect your results as much as like the assembler will at this point. So you're basically giving the assembler almost as good quality data as possible. And so you don't, you're not really going to make anything better by getting it from like 30 to 31 or, 29 to 29 or 30 or whatever. So just because you might see it dip a little bit in the orange for like a few reads, especially towards the end, you'll be fine. If you're in some of the long read sequencing, that is totally a different ballgame. You should definitely look at the documentation for those in terms of their quality, because I know that nanopore data is always getting better and the quality is always being pushed further. And that there's a separate kind of set of pipelines for cleaning those reads up. Okay, now we're going to use Metaspades, and actually for this, it actually is its own app. In fact, if we type in spades, There's hybrid spades, metaspades, and regular spades. We're using metaspades just because we're assembling metagenomic data. And of course, success. Again, let's actually open this again. Okay. So we probably want to use the trimmed paired data and not the raw data. Parameters, just like before, minimum contact length, yeah, 2 KB is probably okay. I would be, in general, unless I have a very specialized data set where I know I'm going to have really small genomes, I don't bother with small contigs. And so, yeah, 2 KB is probably a nice bottom threshold. I know there's some people that have like 500 because they want absolutely everything. And in my opinion, that doesn't really help that much because it just is a lot more effort, a lot more work to filter through all of that downstream. For k-mers, we want to do the same thing, 21, 33, 55, etc. There's also some advanced options that you could look at. I lied. That's all we can change. You can flag assembly only. So if you've already corrected using one of the other tools, for hers, you can do an assembly only run. I have to check. I think it might be regular spades. No, not regular spades. Okay. So yeah, regular spades, you can do single cell or plasmid versions. I don't think, let's check hybrid spades. No, okay. So hybrid spades just adds in, okay. It is cool, hybrid spades. So in hybrid spades, you can have your reads library that you had before. Yes, default, retypes, single or paired end, etc. You can put in long reads here. So these would be your nanopore, spades pipeline options. You can change a couple of these. Of course, this is only available in hybrid spades. You can't get this in the meta spades or regular spades. And then I think, yeah, you can put your k-mer sizes in here. But what I was actually looking for is the DNA source. So if you wanted to run like the RNA version, you would click on, RNA, and that would run RNA spades for you. Okay, yeah, because I opened this too. Okay, so once that's finished, it will generate that object for you, an assembly object, which is basically a bunch of contigs. And then it runs crast on your assemblies. And this isn't super useful. I actually would prefer metacrast because this actually, I think, scans for it's for microbial data where you're assembling like a genome. It's not really designed for metagenomic data. And metacrast actually has a lot of other useful information that really isn't available in regular Quest. So it's nice. You can actually see the total length, the N50, N75, L50s. It's kind of on the smaller end, but it's okay. For viral data, this is actually okay. If this was a microbe, this would actually be awful. So you definitely want to be mindful of what you're, when you're looking at some of these quality assessment tools, like what is your data? Are you working with microbes? Are you working with viruses? And often times you'll find that working with viruses, your distribution is going to be shifted towards the average distribution for viruses. I think for double-stranded DNA phage, it's 50 kb. So having something with an N50, 7.5, isn't bad, especially for one small data set taken from 5+ years ago. Okay, and so now we're going to use VirSorter 2. And this is kind of why I like Cyverse for the visualization aspect, K-base for the visualization, because you can actually give some background on what VirSorter 2 is doing and what it's capable of. And so you can actually see it, VirSorter 2 being compared to different data sets, different identification tools. And clearly it's the latest and greatest tool, so it's going to perform equally or better than its competitors. I do make a note that you should always be cautious when you're examining this kind of benchmarking, because you need to always be mindful about the databases that you're using, the references that you're using. How did they benchmark this? Because no one's going to publish a tool that isn't better than some other tool. It wouldn't make the wouldn't be published if it wasn't as good or better than others. And so you should be suspicious every time there's a new tool out there and really evaluate how they're doing their benchmarking, what kind of databases they're using, because if you're just finding a tool that uses RefSeq and they're like, hey, we have 99% accuracy benchmarked against a viral RefSeq, which is inundated with like a lot of human sequences, then you've basically created a great RefSeq specific viral identification tool. So if you're interested in only viruses that are found in RefSeq, then yeah, that might be a great option. But if you want to start looking at novel viruses or viruses that aren't yet in RefSeq, then you might want to look at tools that perform well for databases that aren't RefSeq, basically, environmental data sets. So VirSorter 2 has a whole bunch of options. VirSorter 2. Yes. So I haven't fully published VirSorter 2, and this is actually why I mentioned it last week, is that not every tool is released like fully. So the beta is actually an opportunity for the developers to get feedback for the community for any tools that are in process. And so you should, as I think I want to paraphrase myself, you should expect beta tools to function, but don't freak out if they don't, because they're beta. I have exhaustively tested VirSorter 2 as a beta app. I'm actually hoping for approval for it to get released to production pretty soon. And so let's see. And so yeah, this will work. I've run it like 100 times. So input config files are the contigs that we just generated. Look through the advanced options. I'm running DramV on this, which is what we went through a little bit last week, and Rory and Michaela went through it 2 weeks ago. And so we want to have outputs so that it's suitable for DramV. And as before, and as I was mentioning before, we can adjust the parameters in order to change our confidences basically in the resulting predictions. So if you want to discard all your short gene sequences with hallmarks, so maybe you have one gene and it only has an RDRP, that's not a whole virus sequence. So maybe you don't want to keep that. Or maybe you want to make sure that you are hitting hallmark genes, you are hitting references. So we'd click on that, and so on and so forth. let's see, I already went over that. Output high confidence. For the purposes of this demonstration, I just clicked on the high confidence viral sequences, mainly because that's what I would use if I'm like processing lots of data. And I don't have to manually curate every single, data set. Viral groups to be included. Instead of typing them all in fact, instead of typing them all in, you can actually see them and add them. So double-stranded DNA phage, if you don't want that, you can do giant viruses, single-stranded DNA, etc., whatever you want. Require them to be annotated so that can reduce false positives. Oh, and something I actually didn't notice, but it's worth mentioning, is that this actually, I've written into kind of like a longer description on each of these. So on some of them at least, how, if you need a little bit more information on each one of these options. And then if you really want a lot of information, you can go through and look at the full documentation that's available in every single app. Okay, so we'll run this, and it generates a, what does it generate? It generates another assembly object. So another kind of like contigs object. Okay, let's see if you. Okay, so question, when I publish my own K-based narrative based on a tutorial, is it okay to keep the explanatory figures citing the original? Yes, I believe so. I would have to ask K-base, but yeah. One great thing about K-base is that it actually has, oh gosh, this is going to be crazy. It actually has what's called provenance. And I'm going to click on this. Come on, come on, come on, K-base, don't fail me. Yes, okay, awesome. Okay, so should go back a little further than this, but so you can actually, and this is actually related to references and citation and all of that. You can click on any of those objects and it will tell you if data was copied from someplace, referenced, or is referencing other data. And so you can actually identify if you were to use someone else's data, you'd be able to not only find them as a reference, but you'd be able to, you'd be able to make your own citation, but also they would see like that you've cited them, basically. And we can see that, that we've gone from the contigs, the Spades contigs, and we've generated a view sorter to kind of assembly object, contigs object. And that, of course, spoiler alert, we have all of the outputs, all of the other tools that basically use this view sorter to output. So you can easily track what happened to all of your data objects. That is something that unfortunately isn't available in Cyberse. I do wish they had that kind of provenance, but they just don't, because just of how they're designed. Okay, so we're getting close. Probably should have started with this and then switched to Cyberse. And then after that, we want to use check fee. Now, check fee is also available in Cyberse, but I totally skipped it. And yeah, this goes over check fee. Everyone should be using check fee to evaluate your virus quality. It's kind of a MUVIG standard. It will give you kind of an overall impression of the quality of your data set. And yes, so we've got 18,000 contigs, and then it annotated 5,400. Yes, identified 5,400. And so CheckV basically uses marker genes to identify whether or not something's viral, to identify, to estimate how complete it is, and to give like a, estimation of, yeah, I guess how complete that genome potentially is. And then it also predicts, depending on your ends, which is sensitive to the assemblers that you're using, and their parameters, to see if it's actually a complete virus. And then I do have highlight that don't expect CheckV to agree 100% with any virus prediction tools. So it's not going to be a perfect match. Do not panic if they don't match. If CheckV finds something that a tool actually wouldn't see that, never mind. So we'd run CheckV. CheckV doesn't have that many options. It has the assembly object, our VirSorter context. This ran quite quickly, actually. And then what CheckV is going to do is give you a table. It was just here. Come on, work with me here. With what it thinks are complete genomes. So it predicted this as a provirus. So it's like, yeah, we guarantee that this is a complete genome. And look at that, it's an 80 kb genome. Goes through completeness for all of the genomes in your data set. Expected lengths, confidence, high confidence, a couple low confidences, medium, high confidence genomes. And this is something that you would report in whatever manuscript paper that you publish. I had X number of high confidence, Y number of medium confidence, etc. Gives you an estimation of the contamination. I don't think there's like any real contamination in some of these, so I wouldn't be too concerned about that. And then an overall, quality assessment. You might find that there are quite a few low and medium quality, but I wouldn't be too concerned because VirSorter 2 is pretty good at finding more novel new viruses. And so CheckV is mainly centered around like, known viruses. I do go over how to annotate them with Proca, but I won't linger too long there. In fact, if we go over, let's see, I know that I'm running out of time. Let's see, sorry. So yeah, we don't have that many options. You only have to change the kingdom to viruses. Genetic code and all that other stuff can stay default as is. Let's open up actually the object that's generated. I just called the object CheckV. And you can actually look through all the features. And while that's loading, in viral groups to be included as the default options include all. No, for KBase, you need to add them. And you'll have to click on each of the options and then click on the drop down and select that option and then use the plus symbol to add a second one. Yeah, so on KBase right now, but VirSorter 2 does not appear. Yes, you'll have to flag this little RB between release and beta, and then it should pop up. But yes, if you've just logged into KBase, and you click on VirSorter 2, oh, I'm sorry. Yes, CheckV says the full viral, so it can just be fed in assembly rather than the VirSorter. Oh, yeah, you could. I can't recall if I think check V, the problem with that, with this question about check V says it's not a full pipeline, it can be fed in assembly rather than the VirSort output. Yeah, you could feed it the whole assembly in Keybase. I think the problem is that check V doesn't generate the result. It doesn't generate any objects. So it's kind of like a dead end, and you can't do anything else with the results. Maybe I can talk to the check-v integrator and see if they can generate an object that's filtered based on the quality. But yeah, and I wouldn't recommend it. I would just recommend what I'm actually doing right now is to identify viruses using a virus identification tool and then run it on check-v. And then, of course, that will generate, Procode will generate this kind of features where you can look at through all of the contigs and whatnot. Okay, we're gonna skip that because that's gonna take forever. Oh, okay. Yeah, you can look through all of these contigs and all the features. I would click on them and you could actually see a visual representation of each contig and the hits that it had. But unfortunately, we are running out of time. There's DRAM-V. Yeah, it's still running. It was canceled when I was trying to run it. But this would give you the DRAM-V kind of results once it's finished running. And then afterward, we'd finally annotate them with vContact2. This is a little bit of the background about vContact2. And vContact2 is one of the few tools that can classify entirely novel viruses. So if they don't match to anything in any database, vContact is probably your only tool. Of course, I'm the developer. I'm one of the developers of vContact2, and so I am a little biased. There are a few other tools that you can use that that are maybe even better than vContact2 for known sequence data. But since we're using an environmental sample and we don't know what could be out there, then we'll use vContact2. And so the configuration options are quite expansive. I wanted to make sure that every user in Keybase could access every single option. The only problem is that no one needs to edit any of these options because the defaults are appropriate. In fact, the only thing you might actually change is the reference database, which is already, it's not an advanced thing. I have an update to this that I'm also trying to push through on Keybase that will have more recent versions of RefSeq. But basically, it's whether or not you want to use oldest versions of RefSeq that the manuscript and all the benchmarking was done on, or if you want to use the more recent ones. Status, result. Okay, so vContact2 will generate a giant table. Yes, sorry, I actually just clicked on this so that it would open up in a window that you can actually see. And it's loading the CSS style sheets now. Okay, awesome. So that's another cool thing you can do with Keybase is that I can actually have HTML and CSS and all of that in here to kind of make it pretty. And unfortunately, I've almost run out of time. I do not want to take anyone past the meeting time. But I do go through and definitely look at this one. When I said I don't want people to, I don't want to have a missing step, this is what I mean. So everything that I would do in order to identify kind of cool and novel viruses is kind of outlined here. Now, the one I wanted to show you was VC277. So one of the things that I like to do is filter and just see how many node, which is an assembly object from Spades. This 545 entries out of the almost 9,000 genomes. That shows me, how many we have. You can filter for the status. So I want clustered. It's live filtering 9,000 things, so it's going to take a second. And then what was that VC? Was it 277? Let's see. I bet it was 277. So one of the challenges, and I need to figure out how to implement this in KBase, I'm talking to the staff, is how to more quickly identify kind of novel viruses. And the only way that I can kind of really explain how to do this is to look for VCs that have both your sequence and a reference sequence in there if you want. That's the only way you're going to be able to find like sequences that match to known viruses. Known references. So for example, if you're filtering through, and you notice that you find a genome, like let's say this is one genome, but apparently its size is 6. Like the cluster, this genus level cluster is 6. I'm like, wait, why is it only showing me one entry? And so if I disable this node or I remove node, you find that my reference, my genome here that was just assembled, that is about 70 KB, is in the same genus, this B tree virus with Pseudomonas. Ignore these two because they're not part of the same VC. But these 6 genomes, I basically assemble it. This is almost definitely a Pseudomonas phage right here. And of course, it's going to be grouping with this particular genus. And so you've now identified potentially me, right here on KBase, have identified a totally new Pseudomonas phage. I would then take this genome and then do some other sort of analysis with it in KBase. I'd be like, oh, let's align it to all of these Pseudomonas phages that we can pull in data from NCBI or JGI or whatever. Yeah, I spent way too long in Cyberse. I apologize. But I do go through this kind of description and explanation. And then, of course, there's 6 other VCs that actually have this kind of feature or characteristic. Yes, feature and characteristics. Oh yeah, and then one of these was actually Prococcus and cyanophage, which arguably the most abundant organisms in the oceans. So we basically, of course, you're going to find ocean viruses. I go through some of the contact options and what all of them mean, because I've got lots and lots of questions about them, so I just answer them here. And then all the references, more reading that you want to do, update, etc. So I do wish that we had more time. There is a multi-hour seminar with KBase that I did earlier this year that goes through in far more detail than I did here. And then finally, if you go to this website, maverickinformatics.readthedocs, I know we're at 1 minute past. All of the apps, all of the tools, I've got hundreds of apps, probably should have opened with that. Hundreds of apps, hundreds of tools that you can use on OSC with instructions, like you can see all the apps on the left here. And then a whole set of viral analysis tools, viral recall, Vibrant, VirSorter, Veridic. All of these are other viral identification tools. So if you're on OSC, just check this website out. And it will have, it has everything Cyberse and KBase has, but more. And then, yes, next week, we'll be talking about genome-based taxonomy and phylogenies, the GTDB TK, which actually is one last thing I wanted to add. in this analysis. So what he'll be talking about next week is something that you actually can do in K-base. Okay, now I'm going to answer some questions. I will stop screen sharing. So sorry that I'm keeping people past. Answered live. Sorry. K-base, yes, answered live. Was it better to assemble a whole genome using just context raw reads against? That's a good question. If you have reference data set that you want to assemble to, or a virus data set that you have, then yeah, you could assemble against a reference genome. Assemble a whole genome. Okay, let me rephrase that. If you have a targeted approach, like you have an expectation that there's going to be certain genomes assembled and you want to improve those assemblies, or you want to, or you know you have a new type of that genome, a new species of that genome, then you could use references to recruit reads and build your scaffolds, and generate like a reference genome, like a pseudo reference genome, and then you could actually assemble against that to try to improve that pseudo novel genome. But typically most people just do a straight raw assembly. Answered live, yes. Better to use, which way is better to assemble a whole genome using contigs and trimming reads together? Well, if you do have quality contigs, high confidence contigs of your genomes, then you could throw those in there as well, alongside the assembly, into the assembly, and trimmed reads together. Yeah, the more data you can provide to the assembler, generally speaking, the better it will perform. Trying to think of a situation where that wouldn't be. Well, once you start getting to like 80 to 100 plus X coverage, then the assemblers start to have problems. So using context, yes. So yeah, up until that point, any amount of confident data you can add in there would be great. And then I think I've got one or two more questions. Oh, yeah, I think polishing is more towards the long read data. I might be speaking outside of my expertise though. I generally, when I hear the term polishing, it's with long read data, which I'm not familiar with. So yes. For reconduct 2, if some genomes cluster together but not have a reference, does that indicate a novel virus? Yes, it does indicate something is novel, or at least it does not map to any of the known or extant references. Yeah, but just be sure when you're doing that to make sure that your new genome, like when you say it's new, like it's a new genus, that you compare it with other data sets or other papers or publications that are in the same environment. Because what we've had happen before is someone says, hey, we've got 100 new genera, and then someone publishes from the exact same environment saying, oh, we have 150 new genera, but you really don't have 150 new ones if someone else had already published, half of them, basically. Also, if a viral genome is low quality and clusters with other low quality, can those low quality be combined to generate a high quality? Yeah, I think conceivably that's possible. In fact, I have seen several instances where vContact will cluster or put in the same VC parts of the same genome that were fragmented because of a hypervariable region that the assemblers just couldn't assemble. So you basically had these two fragments of a low quality fragments of a genome that, yeah, there was some other data that was added in there that actually spanned that hypervariable region. And so that was like a cluster of, at the very least, those 3 genomes. And so they were able to figure out that those 2 genomes would actually be connected, and they had I believe it was 2 different species now for that particular organism, for that virus. I think I've answered almost everything. Excellent. Thank you, everyone. Thank you. I know there's still a lot of open questions, but yes. Hopefully everyone found that informative. So unless we have any more questions, I did go through that really quickly at the end. Okay, yeah. Thank you, everyone, for sticking around. All these, 15 brave souls that 2 hours of me talking, actually 4 hours probably, last week and this week. My one last plea for all of you, and if you know anyone that attended last week that didn't attend this week, if there's any improvements that I can make, I always try to switch up my presentation styles and slides and presenting stuff, occasionally, and I just try new things. So if there's something that worked for you, let me know. And if there's something that didn't work for you, also let me know. Because I really do, I write notes and feedback from users constantly, because I want to make sure that no one is left behind. And going through quickly is a great way to lose people. So yeah, let me know. I put my email in there. This is recorded, so just go through and grab that email.